Okay, we're recording. Future meetings, we're going to be meeting on February 21st at Tanner Research. And Robert Kirshner, who's heading up the 30-meter telescope project, is going to be presenting on what they're doing, and they're doing it in Monrovia, in part at least. So that's next on the February 21st. We normally meet once a month for a San Gabriel Valley Tech Mixer, which is like 5 to 7 on Friday. Everyone's welcome to come and just hang out and talk shop. And we may not meet in January, but maybe in February, and we need to find a new location because Wing Walker, where we used to go, is apparently closed. So we'll be getting the word out. If you look on LinkedIn, if you join the LinkedIn group, or if you have Instagram and follow us, you should get the information about it. It doesn't come out on Eventbrite. If you have any ideas for future programs or comments on this program, let us know. It's email@mediatech.org. And after the program, stick around, and when we get a critical mass, we'll go over to Hop Secret Brewing Company to continue our conversation. If you want to join us with that, it's at 162 West Pomona Avenue, which is by the Gold Line station. I haven't been there before, but somewhere over there on Pomona Avenue, which I definitely know what that is. Just some background quickly on Medea. We're a 501c3 nonprofit educational organization. Our mission is to have programs like this to ensure the technical, scientific, and industrial workforce are connected and informed on entrepreneurship, business finance and operations, technology development and commercialization, all with the goal of bringing people together to collaborate and network. So with that, I will introduce our presenter, Dr. Mark Baum. He is the president, senior faculty, and founder of OCREST, which OCREST is an innovative chemistry and biology research and education center located here in Monrovia. Dr. Baum earned his PhD in organic chemistry from the Imperial College London. For the past 20 years, he's been active in the development of sustained release drug delivery systems for HIV pre-exposure prophylaxis, including injectable, implantable, and topical strategies. His principal research interests include development of innovative and practical long-acting biomedical technologies to combat infectious diseases, pharmacology of antiviral agents for HIV prophylaxis, drug discovery of novel antimicrobial peptides and peptide drug conjugates, and microbial dynamics in specialized niches. So please welcome Dr. Baum.
Thank you, Bruce. Thank you, Bruce, for allowing me to be here today, and Paul for putting today's session together. I'm really glad to be back with Medea. It's been a while, and I'm really excited to see this high turnout here today. So this is really wonderful. I like speaking from one person or 200 people, equally the same, but it's always gratifying to see a bunch of faces. So this is wonderful. Thank you for being here. So the talk that I've prepared today is the whole point here is to take you through a journey, as the title says. So this is going to take a little while. I had very sort of ambiguous responses to how much time I had. So I'm going to take as much time as I want. And if you don't like it, the door is there, and you can go to tour while I finish up. But I'm hopefully going to keep you engaged and take you through a discovery process, not just of new molecular entities, new technologies, and new results from clinical trials that hopefully will string it all together and make sense in an exciting story. So this little arrow works. That doesn't. So this one doesn't work either. Got all the technology issues here today. Can't get the pointer back either. Here he is. All right. So I am reintroducing OCREST because I looked it up, and it's actually nearly to the day 9 years since I last presented at Medea. It's crazy, right? It's been that long. Yeah, absolutely.
Yes, I remember that.
So in early 2015 is when I presented last, and we had just purchased this building, and we were planning to move in. That took a little longer than anticipated, but we're here now, and we're very, very excited to be part of the Monrovia community for this long, and hopefully many, many more years to come. As Bruce alluded to in his kind introduction, we are a nonprofit organization, and our fundamental mission is kind of bifurcated. On one hand, we are doing cutting edge, environmentally and medically relevant research that will have hopefully a positive impact on society. And at the same time, we're using that exciting research to bring in the next generation of scientists and train them and help them learn how to be researchers. Because research is something that will get you hooked on science more often than not. And unfortunately, a lot of young future scientists don't get the opportunity to do research until much later in their careers. And at that point, they may already have lost interest in pursuing a career in science. So a lot of people don't know that this could be something for them. And we do this in a very sort of, as we call it, academic entrepreneurship type environment. We're very small, as you can see, and yet we have staying power. We've been here for over 25 years, and that is exciting. We own this set of buildings here, and hopefully another one in the not too distant future. And that gives us, again, sustainability for the foreseeable future. One of the fundamental tenets for us to be successful is to collaborate with others, and you'll see this throughout my talk. And that is what allows us to really expand and amplify what we're doing. So my focus today is going to be on the biomedical part of the research that we do. I'm sorry, I know we have one person here from Monrovia High School is interested in the environmental. That'll be for another day. But hopefully the biomedical part will be of interest as well. So when you start doing biomedical research and you're looking at platform technologies, potentially drugs as well, you have to have some sort of focus. You can't just do everything as much as we'd like to. And infectious diseases is something that we are especially focused on, interested in, because infectious diseases are a major problem worldwide. You're all wearing masks here today, and I thank you for that. That is because we still have a SARS-CoV-2 COVID pandemic. It's gotten much better over the years, but we have a major flare-up at the moment, and that's still a problem. It's still costing people lives. It's still getting people sick. It's making people lose work time. It has a major economic impact. HIV is still a problem, and then there's all the major other big infectious diseases. So we have our work cut out for us for many years to come. And all the bullets that you see, I'm not going to read them all out to you. They're all different aspects of what we do here at Oak Crest, which is really remarkable because we are a small institute with less than 20 personnel, and yet we're able to do drug discovery, which you'll hear about in a minute. We're able to do new drug delivery technology development, and we can do manufacturing in-house for clinical trials. We do regulatory work to get us into the clinical trial. We actually oversee and collaborate with clinical trials. We do clinical studies here at Oak Crest. And we also involve all the different researchers at different levels in all aspects of what we do here. So this is pretty comprehensive. My focus today is going to be on HIV, as Bruce said. And it's really something that we've largely forgotten about here in the US because we think it's under control, but it's not. HIV is actually getting worse in certain parts of the US. And in certain parts of the world, it is still a very problematic disease. It's still an epidemic that we're having a very hard time controlling. UNAIDS has set very aggressive targets, the 95, 95, 95. I'm not going to, again, read the slide for you. You can do that yourself. But basically, these are not being met. We're having some major issues attaining these targets. And one of the main reasons for that is that we are unable to reach adolescent girls and young women in sub-Saharan Africa. Within the 1 hour that you're going to be here with me today, 24 women, young girls and women will be infected with HIV on average. So these are staggering statistics. And this just keeps on going. And there is no apparent end in sight. So the heterogeneous nature of the HIV epidemic here in the United States, it's more men who have sex with men who contract HIV. Women are starting to get more at risk as well. And in sub-Saharan Africa, which is one of the big areas where HIV transmission is still occurring, is adolescent girls and young women. So you now say, okay, I want to embark on solving this problem. I want to contribute to helping people and doing something about it. what does the landscape look like? So this is kind of the flow chart of what it takes to start from ground zero. Like today, I have an idea, I think I may be able to design this new compound and take it all the way through to a commercial product. So there are a number of steps involved, quick key steps, which are highlighted here. At the bottom, in the little sort of pale squares, you can kind of see some of the pitfalls where a lot of these things usually fail. So where we as academics would fit in is that blue box, the basic science, where we're looking for a specific target, or maybe not. We're trying to identify a lead compound. We do in vitro studies to support the selection. We look at overcoming certain toxicity issues, some potency issues, and then we move to the next phase, the orange one, where we're doing preclinical. So these are early stage animal studies, looking at multiple species, seeing if the drugs are safe, do they work, and can we get the funding to then do these really expensive animal studies that are done under what's called good laboratory practice, GLP, which basically is a sort of standard that you're set that is very rigorous, which is also very expensive. So at that point, a lot of the drugs fail, but those that succeed, they go on to the next phase, which is tech transfer, something that Bruce knows a lot about, manufacturing, regulatory, etc. And then you can finally, after spending millions of dollars, get into your first clinical trial, and there a lot of things can fail before you even get to the chance of presenting your case to FDA for approval. So I'm going to start with the drug and not worry about how it's delivered at this point. And I'm going to talk to you a little bit about peptides. How many of you know what a peptide is? Okay, decent number. How many of you know what a protein is? There you go, much better. So a peptide is a tiny little piece of a protein. And a protein is made of multiple amino acids, hundreds, if not thousands, that are all tied together one at a time. With a peptide, you're looking at probably less than 50, sometimes a little over 50. Around 50 is kind of the cutoff of what defines a peptide. So it's a much smaller piece of a protein. And these have not really, other than a few, which I'll talk about in a second, peptides have been very slow to becoming commonplace in terms of our drugs. They had enormous potential. They used to be expensive to manufacture. And as a typical problem with peptides is that you can't take them orally. They have to be given by injection. And that, of course, is an additional burden that a lot of people don't want. However, as of 2022, we had about 80 drugs that were approved as peptides. And if you watch TV, you've probably seen a few commercials with some of these drugs. The sales are getting really quite impressive at over 70 billion in 2019. And there's a whole bunch of peptides that are in clinical development, and those numbers have doubled in over 10 years. So it's really a growing area. The first peptide has been around for a long time. It's insulin. I'm sure you've all heard about that one. And that has been approved in 1923. So that seems, well, you just said that peptides is kind of a new thing. Well, insulin is kind of the exception. And then it's taken much longer for the next generation of peptides to come along. And there's some really interesting ones out there. Exenatide, that you may have heard of for type 2 diabetes, was actually originally extracted from the saliva of the Gila monster, which is like a large lizard that lives in the southwestern United States. So this one came from nature. A lot of peptides do. I said that peptides usually are given by injection. Well, in 2019, so just a few years ago, oral semaglutide, which is also for diabetes, was approved by the FDA. That was a major breakthrough because now we can start to have strategies for delivering peptides orally, which is really wonderful. And then another example is a sea snail has a venom that contains a peptide that was isolated and then became FDA approved for managing pain. And then we have also got a peptide that is a fusion inhibitor, so it prevents HIV from getting into the immune cells, that became approved in 2003. So there's actually an HIV peptide, anti-HIV peptide that is approved by the FDA. So as I said, our interest is in infectious diseases. So let's talk a little bit about antimicrobial peptides. Well, they're very common in nature, and they're very part of what's called the innate immune system, which is basically the opposite of antibodies. It's a general broad immune system that targets everything, that is multifunctional, and that is not specific to one agent. And that represents an opportunity because there's already a rich history of those around, and a lot of them are known, but a lot of them are not particularly good if you want to purpose them for a specific organism, which gives us an entry point to re-engineer these peptides and make them work for new applications. So for example, melittin, which you may or may not have heard of, shown in the top corner, is a trimer, peptide trimer, and actually is the main component of bee venom, but it's also really good as an antiviral. It works against HIV, and more recently it's been shown to work against SARS-CoV-2. So there's a lot of opportunity here to mine nature for peptides and then, as I said, engineer them for specific applications. What's cool about these, because they are broadly antiviral, broadly antimicrobial, they don't actually target a specific receptor, which is kind of unusual for drug discovery because you usually are looking for targeting a receptor, but here they don't. So you're targeting certain features on the surface of a virus, maybe it's chemical features or structural features, and that really helps in many ways have a broader activity. You're less sensitive to changes, you probably have less toxicity in certain cases, but on the other hand, it makes things a little bit more complicated because the mode of action of some of these compounds is quite complex and can be made up of multiple mechanisms. So here at OCREST, we have several people in the audience today, online and in person, who actually are doing peptide discovery at OCREST. I never thought I would say this. When we moved in here 10 years ago, I probably wouldn't have said that, because that has usually been an expensive process, time-consuming process, very inefficient process, and only the really big pharma companies could really do this in an efficient manner. But over the last 15 years or so, as I like to say, peptide discovery has been democratized. These instruments have become much cheaper, and there are tools that are available to us that allow us, even OCREST, as a small organization, to do this in-house. So this workflow, I'm not going to go into detail because others will be here all night, but basically, you have a peptide synthesizer where you can make one, or depending on the synthesizer, up to 96 peptides in one shot. So 96 different peptides in one shot. And then there's a whole workup process where you may do some preliminary purification. You identify the peptide, that's what Amalia and Joe would be doing here, and make sure that you have the right, you've made what you think you've made. Then there's a purification step, and then you can slide the peptide into a whole bunch of different assays. And I listed the 5 key assays here if you're doing a vaginal HIV prevention product, for example. So we do all of this at OCREST. We have all these instruments here downstairs, and I'm sure somebody will be happy to show them to you later if you're still awake, and take you for a tour and really take you through the workflow. It's absolutely fascinating that you can assemble these things like Legos, essentially, because you have 20 natural amino acids and hundreds, if not thousands, of unnatural amino acids, and they're all like Lego blocks. And you just program the instrument to assemble them for you, and then after that, the magic happens, and then you've got to start to apply your creativity. It doesn't always work, so you have to be able to troubleshoot. But basically, it's a beautiful process that once it reaches this threshold of pricing, can be done at a small organization. So what are we working on here at OCREST? Well, on the left side, you'll see what's called an alpha helical peptide. It's kind of like a corkscrew, and it is a peptide that was originally developed for preventing or treating hepatitis C. Didn't work out, was abandoned. And like I said, we picked it off from the dumpster, repurposed it, and spent a lot of work making a library of over 300 peptides based on that original sequence to develop a peptide that is active against HIV, and it's optimized and stable for vaginal drug delivery. This is now moving into a clinical trial probably in a year and a half from now. So we're already in that very expensive phase where we're trying to convince FDA that this is safe to put into people. So this has moved along very, very quickly in the last 2 to 3 years. So very, very proud of this project. It's been definitely an interesting journey. The other one is a little bit earlier on. It's a more exotic peptide. It's a circular peptide, so it's cyclic, and it's a macrocycle. It's a large cyclic molecule, and it has a ladder of 3 bridging species that tie the whole thing together and give it really exceptional stability. That's the original, we call it the progenitor, the starting compound that we're working with. And that's a very, very interesting compound where I can be talking with you again for hours on this thing alone. We did not discover this. This was discovered by a team at UCLA in 2002. But basically, it is one of these innate immune components called defensin. This particular group of defensins is called beta defensins. We actually have the genes to make them, but we don't express them. So they're basically dead genes in our system. They're turned off. But certain types of the lesser primates actually do make them, and that's how the whole thing got started. And then we were able to express the genes from the humans to make the human version, and so on and so forth. We're working on a simplified version of this. These are a little bit complex to manufacture, which is not something you want when it be cheap. And so we're working on a derivative version of that, and we're in the early stages of that. So here, very quickly, I don't want to dwell too much on this. Here are some of the results, just to show you that I'm not just talking, that there is actually data from our linear peptide that I mentioned earlier, the one that's going into clinical trial. The green graph shows basically that as you increase the concentration to the right of the peptide, your efficacy in preventing HIV infection of immune cells goes up until it reaches 100%. And then when you do the same thing, look at the toxicity to the cells, if you look at the x-axis at the bottom, you will see that the numbers are much bigger, which means that the toxicity needs a lot more drug for it to become toxic. So we have what's called a very good therapeutic index, which is essentially a trade-off between efficacy and toxicity. When you take the compound from cells, from an in vitro system, Petri dish, and put it into animals, humanized mice. So these are mice that have a human immune system, that are grafted with a human immune system, and can be infected with HIV, the human virus. You can see that you have a very similar dose-response relationship in orange. And the numbers are slightly different because obviously a mouse is not a cell, and a cell is not a mouse. And when you do a similar study in mice against herpes, HSV, the compound also is effective. In fact, it is as effective as one of the key drugs that is still used today to treat herpes. And that is a different kind of plot. It's called a survival plot. So you can see the gray is representative of the mice as they become infected over time. And at the end, after about 9 days, all the mice are infected. There's 0 mice left that are uninfected, whereas the purple plot is mice that are being dosed with our peptide vaginal. This is all vaginal. And you can see that only a couple of mice have become infected, and that's late in the study. So this is a very good way of showing efficacy. What we also want to do is make sure that our peptides, which are biomolecules, right? So there's a lot of mechanisms in your body to break them up, that they don't fall apart. And that took a lot of effort. A lot of people here know that we spent many months sweating over this, but we now have engineered this peptide with some clever little tricks for it to be very, very stable in vaginal fluid. And that's what that last plot shows. Okay, so I've convinced you now we have this fantastic drug that is going to solve the HIV prevention crisis in sub-Saharan Africa. So what do we do with it? We've got to deliver it somehow to the vagina, right? And I said to you that most peptides aren't orally bioavailable, so you're not going to be convincing adolescent girls and young women in sub-Saharan Africa to inject themselves every day with the peptide. So what do we do? So we're going to talk about drug delivery. And the drug delivery, I like to refer to it as the black sheep of the drug development process because it's not particularly exciting to most people. It's a pill or an injection, right? How fascinating scientifically can that be? But there's a lot more to it than a cream, a pill, an injection. There's what we call long-acting drug delivery. And that's a lot more exciting scientifically. It's basically saying that people don't want to take their medications every day. They want to have something that they either inject or implant or wear that protects them, in this case from HIV or from getting pregnant, for months at a time, maybe years at a time. And there we have a lot of opportunities to make an impact. So now that we have this really cool peptide, how do we deliver it in a long-acting format? Well, you really have two different main options. You can deliver it systemically, which means you deliver it into the bloodstream. You can do this with an injectable system, not an injection that you would have every day, but one injection that you may only have to have once every 2 months, every 6 months, or an implantable system. And you can see here, there's the examples of FDA-approved products that do both. You have the Implanon that I'm showing here, which is this little matchstick-like device, which has been around for a long time for contraception. It gets put into the upper arm. And then you have this injection here, which just became approved by the FDA a few years ago for preventing HIV. It's called Apretude. It's a HIV product that is actually being used in the United States. You may have heard about Cabenuva on TV. This is kind of the same thing. Cabenuva is for treatment, and it's 2 drugs. This is prevention, and it's 1 drug. But it's the same technology. And then we have the topical drug delivery platforms where you can have patches that you put on your arm. You might have heard of these microneedle patches. And then you have intravaginal rings, which the NuvaRing showing up here is a very well-known example of that. All right. So in terms of HIV prevention, there's a long history of activity in this field. The vaginal ring was actually originally filed as a patent application in 1968. Patent was granted in 1970. So vaginal rings have been around forever, right? They're older than I am. Well, not quite, nearly. And it took a while before the vaginal ring was recognized as a valuable platform for HIV prevention. And in 2005, there were some seminal studies by Carl Mulliken in Belfast, in Ireland, and he developed a very simple vaginal ring, which I'm showing at the top here. These pictures are from one of his publications, delivering an antiretroviral drug called Bepivarin. And jumping forward, the Bepivarin ring from 2005, so this gives you an example of how long this takes. From 2005 to 2016 was the development of this ring. And finally, in 2016, it was tested for real in 2 huge clinical trials for efficacy, both of them in Africa. And in 2023, the Bepivarin ring had been or was being approved and authorized in 11 countries in Africa. And it's already available in 6. So this is a big breakthrough for HIV prevention, but it's taken a long, long time. Our approach is a little different. We developed what's called the POD IVR, which is a unmedicated ring that contains little white tablets. And each one of these tablets is a drug delivery system. It's a very simple device. The tablets are compacted drug with or without additives. You have a layer around the pill, just like a lot of other pills. And then the drug releases through relatively large channels that are engineered into the ring itself. And by changing the number of channels, how big they are, the coating on the drug tablet, we can control how quickly or how slowly this ring delivers its cargo. Here's just some examples of different entities that we've delivered with this technology. We've delivered antiretroviral drugs all the way through to probiotic vaginal bacteria. So I said entities because I didn't want to say compounds, because we actually also delivered live organisms. I'm going to jump through some data here. I'm not going to stay very long on this because we don't have time today. But basically, just to show you that we've done a lot of things with these that are very unconventional. We were able to, under John's leadership, deliver an antibody against HIV, specifically against HIV, a monoclonal antibody called VRC01, developed at NIH, and deliver that from a vaginal ring. That had not really been done before, at least not in a controlled fashion like we did. And we evaluated this ring in macaques. This graph just basically shows that we're able to maintain, not particularly well, this was an early version. We can do high, low, and mid release of the antibody into vaginal fluids, which is why it's active in macaques. And then John and his team showed that, and I circled that data, within 1 hour of placing the ring, you already have really high drug concentrations vaginally. So this is something that really is an on-demand product. And that next generation of the ring that John and his team developed actually delivered reasonable levels of drugs for 3 weeks. Again, not phenomenal control at this point, but it showed a lot of promise. So we took a step back and said, okay, well, there's oral Truvada, which is a combination of these 2 drugs that you see at the top here, TDF and FTC. The 2 antiretroviral drugs have been around for a long time. And that is an FDA-approved oral regimen for preventing HIV. It was the first one. We thought, deliver both drugs vaginally, that should work too, right? So we developed a ring, again, based on this POD IVR platform, and made small macaque-sized rings, non-human primates, and they can be infected with a hybrid virus what's called SHIV. It's kind of a monkey version of HIV. But it has a lot of features of HIV. And we basically did an efficacy study with the CDC using these rings, and it's 16 challenges with the virus every week with these rings in place. So that's what that graphic shows. And here's another one of these survival plots. You can see that the gray pattern shows the monkeys becoming infected gradually. A lot of these are historical controls. We actually did not use live monkeys. We only used 4 in that group. But the purple part is the beautiful part where not a single one of the monkeys with our ring became infected with the virus. So we showed this works. So we then took it a step further and we did a clinical trial with that basic ring technology. And we're able to gradually add, and this is one of the beauties of that pod IVR technology. We started with just TDF, one of the 2 drugs in that Truvada combination. We then added a second drug, and then we added a third drug that is totally different, also an antiretroviral drug. So this is a clinical trial in humans, in women, that we did the regulatory process to get permission from the FDA to do this trial, and we, with our collaborators, ran the trial. So this is a pretty big accomplishment for a small nonprofit. And basically, you had these 6 women use the ring, then switch to the second ring, then switch to the third ring. So it's a very elegant study. And very quickly, I just want to show you that we measured the drug concentrations, the key drug concentrations for all 3 rings. That's what you see here at the bottom. And as you add a drug, you add a new part to the plot. So the last plot has 4 drugs. 1 is a metabolite. So 3 drugs plus the metabolite. And it shows that as you add the drugs, you get a new part of the plot. But the old drugs, the blue one, for example, that's there in all of them, they're all pretty much the same, which is what you'd expect. You're just adding a new drug. You shouldn't really be impacting the other drug. That's in the vaginal tissues. And that's the compartment where you're protecting from HIV infection in women, vaginally. One of our collaborators, Peter Anton, who's faculty here at Oak Crest as well, he's been involved in HIV research for a long time. He said, you know what, while you're at it, see if you can collect rectal fluids from these women. And like, rectal fluids. I said, dude, it's a vaginal ring. Go to the vagina. Yes, but you're doing this anyway. If they consent, just collect rectal fluids. You never know. All right. And I expected to see nothing in the rectal fluids. And I was wrong. Basically, what's fascinating is we can actually see the blue drug, tenofovir, in the rectal fluids in all 3 cases, except that in the case when you add that third drug, Maraviroc, you barely see any. What's even more dramatic is FTC and emtricitabine, the second drug from Truvada, the red one. It's there in really high concentrations in the double ring, but the moment you add that third drug, the orange one, it decreases dramatically, and that's statistically significant. And that's a beautiful example of drug-drug interactions. You very rarely see it as pronounced as this clinically. So we showed something that I was wrong yet again, and that we had drug-drug interactions. All right, I'm going to take you through the final part of the journey. And I'm glad to see that there's virtually nobody here who's asleep. So that's really, there's a couple of you guys that have your eyes closed, but I know you're still paying attention.
It's the wind.
Exactly. So we took a step back after we did pretty much all we could do with the pod IVR. You know, we did clinical trials, we delivered vaginal bacteria. We thought, okay, you know what, now's a good time to reboot and see, is that really the best way to do things before we really start to invest in going all the way into these bigger clinical trials. And as I said, Duncan's patents, and I really love this image. So at the bottom here, these 2 figures, the 2 black and white ones, these are actually taken, and this is actually signed by the attorney in 1968. I think it's awesome to have this. And these are 2 images from that original patent. And as you can see from the figure 1 and the one that's being held here, this is the dapivirine ring that has been approved last year. And the NuvaRing down here, which is being used by millions of women, they look all the same. That's over 55 years ago. So Duncan was brilliant, but we really haven't done much with it since then. Even had a spring in one of his rings that nobody's using today. So that was kind of, it was way ahead of his time. Our pod IVR is different, but it's still using the same shape. So is there something better that we can do that's more technologically exciting and has more potential in terms of vaginal drug delivery? So John and his team started off with some sketches and a sort of space design here with sort of looks like a spaceship at the top right there. And that was version 1. And basically the idea was don't settle for the donut shape. Use a space in the middle because that's just wasted space. When the ring is in the vagina, actually, it folds along the vagina. It's kind of like a sort of slightly open taco. And it rests against the vaginal wall. It does not sit on the cervix like a cervical cap. That's a misnomer. So that all that space in the middle of the ring is basically wasted space that you could do something with it. So we started off with the concept of having 2 drug delivery cassettes in that space and try to figure out what's the best way of doing it. So generation 1 didn't really make very good use of that space. The ring was not particularly flexible in certain areas that could be problematic. So we went to a round cassette and John and one of our mechanical engineers came up with this really nice design of how you could have 2 interlocking parts of the cassette, which is that blue piece on the side here. This is an open cavity without a cassette. Each cassette could contain 1 or maybe 2 drugs, and you could deliver from that. But again, we felt we could do better and use make better use of the space. And then we would want to seal a membrane. You can barely see it here where these little holes are. There would be a membrane that would be sealed in there. That gives us additional ways of controlling the drug release. And that was difficult to achieve with this design. So the designs evolved, and I'm just really showing you these just to see how the evolution happened. We're talking about a journey here. And eventually we arrived to this design. This design is actually the design that we're going to proceed with in the clinical trial for our peptide. And this design is very cool because it has a reservoir, which is the pink part here that I'm showing, that contains the drug. So it can be solid drug, but more than likely it'll be a paste, easier to administer to the device when you're manufacturing it. It has a bottom cap here, shown in teal, that basically holds the drug. It has a white membrane, and it has a cap that seals over the membrane. The whole thing pops together. We can have it with sort of little pins that guide the assembly. You can have different iterations of this, but this is the overall concept. It may look complicated, but it's actually really cheap to make. It's really easy to assemble, and it is very scalable from a pharmaceutical manufacturing perspective. From a scientific perspective, the one thing that's really cool about it is now that you have an actual physical membrane that is fairly large, that fits nicely inside that cassette, you can start to use really exotic and cool technologies to as membranes. So for example, we came up with this sort of, we call it the egg carton or the waffle design, where basically you have these peaks and valleys in this grid. And here's an actual membrane that we made and evaluated, that you can actually make the little waffle squares bigger or smaller, deeper, and make different materials. And you have an inordinate amount of degrees of freedom that you can control. You can go with nanostructured membranes, something that John might be interested in. There's a number of groups that are working on these. They're very cool technologies. So we took this new ring design that I was talking about, and a lot of people, when they saw it, they thought, "Oh my God, this is way too big. This is never going to work. Women are going to be freaking out when they see this." And so we decided, okay, well, let's try it in women and see what they say. So this is an unmedicated ring that we started off with, one of the earlier versions. And with our collaborators in Texas, we came up with this really innovative way of testing the device, taking user feedback, changing the device, and putting it back, cycling it back into the testing cycle. So basically, we had an initial set of prototypes in that first square. We tested them in sheep. Sheep have vaginas that are very similar to humans in terms of size, anatomy. Also from a safety perspective, the epithelium is very similarly structured. So we evaluate them in sheep. Now they can't really tell us if they like it or not. They can't really take them out very easily with their hooves. So it wasn't a perfect evaluation, but it was a first start. The beautiful thing is our collaborator, Kathy Vincent in Texas, she is actually an OBGYN, but she also runs the sheep studies. So she actually deals with women and with sheep. So she can translate a lot of the results from the sheep studies and say, this is how it would be in women. So based on the evaluations in sheep, we made some changes, we rejected some designs, and then we moved to a clinical study where in clinic, women wore the ring for about an hour after placing it themselves. They would be jumping up and down chairs, running around, coughing, etc., to see if they could feel the ring or if it would pop out, and then they would remove it themselves. We also had transvaginal ultrasound, which is essentially a sort of phallic-like device that gets put into the vagina to do in-vaginal ultrasound imaging to see where the ring was, but it also kind of simulated sex as well. So you can see, do the women feel the ring during sex? This would be an indicator. If they have pain when you do it, it's problematic. And again, that allowed us to refine some very subtle design changes to the ring that eventually resulted in a winner, which is actually, as of a month or two from now, going to be evaluated in this large study down here, which I'm showing as Matrix 003. It's a 28-day clinical study with 5 different sites, 4 sites in Africa, 1 site in the US, 100 women participating. And they're going to try 2 different rings, same design, but different hardness silicone. And hopefully we'll find out which one they prefer, if they prefer one. And that's the one we're going to go forward with. So this is very exciting times on this ring. And this is what the ring looks like. I've just summarized a lot of what you see here, so we're not going to have to go over it again. But that is kind of our winner. And you can see that when you fold it like a taco, that's the way it gets placed vaginally. And that hinge area here that you see is very important for removal of the ring, because the women go in with a pinky, grab the hinge, pull it out, and it's very easy to place and remove. In fact, a lot of the women prefer it to Nuva rings. Half of our participants had used a vaginal ring before, and they really liked it. So I'm nearly done. I want to show you kind of our flagship program at the moment, or one of our major programs at the moment, which we call the One Ring. Not our name, somebody else came up with it. But basically, the ring that you see on the left-hand side is exactly what these placebo rings look like. And it's a really, really innovative product. It's a dual-purpose prevention product. It contains our linear peptide that you heard about way at the beginning of the talk. As an anti-HIV agent, it potentially can also prevent herpes transmission, HPV transmission. So it is a multiviral, but we're focusing on HIV. But in combination, we're delivering a non-hormonal contraceptive. So this is a contraceptive that blocks sperm from swimming. And you can see here 2 images. The first one at the bottom is sperm with just vehicle, not the small molecule Sac inhibitor. And you can see how the tail of the sperm is beating. That's what those colors are showing. So it's moving. The moment it's exposed to very, very low concentrations of this non-hormonal contraceptive, it becomes non-molecule. It's essentially dead in its tracks. Sperm is still alive, but it's not moving, not going anywhere. Really innovative compound, avoids all the problems with hormonal contraception, does not need to be systemic. It works locally in the vagina, just like our peptide, which is a shield against the virus, as I'm showing pictorially here. Yes, I'm nearly done. So this is a really innovative combination of drugs that has not really been tried before. And you would say, well, why a contraceptive? The adolescent girls and young women in sub-Saharan Africa are much more worried about getting pregnant than getting HIV. So the contraceptive is a carrot for them to use the device. They use the device to not become pregnant, at the same time they're protected from getting HIV, which is really our main goal. So on top of having 2 new drugs, these are 2 new molecular entities. These are not approved drugs. We also have a new device, a new ring device. So you can imagine that the regulatory part of this is nightmarish. And I am not aware of another product like this. So mostly you have a new device and a new drug, but not 2 new drugs and a new device. So we're spending a lot of money, not ours, thankfully, but people have a lot of faith that this is going to be a viable product. And we'll find out when we do our first in-human clinical trial in 2025 to see how well this will really work. But this is a really exciting project. So you can see now we've come all the way from the discovery phase, where we read an interesting paper about a hepatitis C drug that failed, repurposed it in-house in our little peptide facility, did drug discovery, device development, and we have a beautiful device now with the help from our collaborators at Cornell with their non-hormonal contraceptive. Could be a major game changer in the HIV prevention world. So as I said at the very beginning, we would be nowhere without collaborations, but most of all, we would never be anywhere without a fantastic staff, which is some of the senior scientists are listed at the top. And then there's too many researchers over the years that did a lot of this work that I couldn't list them all here, unfortunately, but we have a phenomenal team of past and present researchers here at Oakcrest, some high school kids, community college kids, and all the way through to PhDs, who all work together to develop and test all these technologies and hypotheses. A list of collaborators, and then of course the funding from NIH and USAID. So this is it. I'm here for questions if you have any, or if you want to go on the tour, there's still some time before we do this.
I do have a question from online. Vishnu is wondering about safety studies. Have you conducted or not shown here safety studies?
Yes, so that's an extremely important question, because to get into your first in human clinical trial, the only thing that matters is safety. Efficacy is something you test much later, but obviously you have to have a product that might be efficacious one day. Otherwise, what's the point? But safety, protecting the volunteers who are taking part in the clinical trials is of most paramount importance. So this is a very important question. And yes, we have done a lot, millions of dollars worth of safety studies, starting in mice, then in rats, then in dogs, and in sheep. So we are about 2/3 of the way there to having all the safety studies that we need to get FDA to let us go into humans.
Okay. Question online.
Felipe.
What will it mean for you if you do succeed in getting the FDA to approve this? What will it do for Oakcrest?
That's a tricky one, right? So number one, I mean, I know this, you may not believe me when I tell you this, maybe you do, you've known me long enough. We're not doing this for the money, because if we were, we would not be developing a product for Sub-Saharan Africa. There's no money to be made there. This is a humanitarian project where obviously, as a nonprofit, that's partly what our mission is all about. So what's in it for Oakcrest? If we can get to the point where we have successful early stage studies, trials, and probably will be involved in the phase 2 safety study as well. If we get to that point and show the ring is safe in hundreds of women, then we would probably partner with someone else to take it over. It doesn't mean that we would still not be involved, we probably would be, but there are either for-profit, Merck, for example, is a pharmaceutical company, or it could be a nonprofit organization that does these larger clinical trials together with, say, NIH, and get it all the way to the point that it becomes an approved product. Once it becomes an approved product, it would probably be taken over by somebody else for manufacturing and distribution in all the various countries where it's needed. And so for Oakcrest at that point, other than immense satisfaction, there would probably be not much else there. And of course, visibility and recognition. Although at that point, you know, unfortunately, like Carl Malcolm, the guy who developed the Depivir ring that's approved, most people don't even know that he developed it, because the nonprofit who took it over and got it to approval is taking all the credit. So it's not about credit, but that's part of it. Now, on the flip side, if the ring is active against herpes, which it is, and can be a contraceptive, the same exact ring could be a very good product for the developed world. So there we would be looking for a licensing deal, et cetera, et cetera. We got to pay you, right? And so, so we would try and organize something like that, and also the licensing deal and having it be a successful product in the developed world would help fund some of the work in the developing world. So that's a long answer, but it's an important question that one has to really think about.
Thank you.
So we were wondering if the new design extends the amount of time it can stay without reintroducing new drug, and is it a whole new ring, or do you actually just change out the membrane and the drug?
Really good questions. So yes, you can see that you're a student in observing that those gray cassettes that you see there on the screen, obviously much bigger than the normal vaginal ring. So it can, you're absolutely right, it can contain a lot more drug than the normal ring, So in theory, you can go much longer than a normal ring. So from a drug potency perspective, you can probably go 3 months with one ring, maybe even longer. It's a regulatory nightmare to start with. You would start with a 1 month ring that you change every month, just like the NuvaRing, which is a 3-week ring actually. But because women would still have menses and have completely normal hormonal cycling, they could use the ring for the whole month and then swap it out after the period, for example. There is a contraceptive ring that's out there right now that's approved for 1 year, but a lot of women don't want to use it for a year, and especially in the focus groups that have been had in sub-Saharan Africa, a lot of women don't feel clean if the ring has been in during menses, for example. So most likely the product would be a 1 month ring that you would then replace, but a woman could get say, you know, a kit of 6 or 12 rings to take home, and then just swap them out every month and then throw them away. They wouldn't have to go back to the clinic to get them.
Yeah, do you have a good idea of what's causing the metabolism in vivo, and is there some kind of like endogenous enzyme that you could like inhibit to therefore dose less, and like adding another drug to the ring?
Yeah, I mean, there are already a whole host of HIV drugs, you know, that are approved or are on the verge of approval that have all sorts of different mechanisms against HIV, and that do, yes, indeed, interfere with different facets of the HIV replication life cycle. The problem with that is that those drugs have to be in the cell where HIV is replicating. HIV can only replicate in certain types of immune cells, and so you have to get the drug from the ring through the vaginal fluid into the vaginal epithelium, into the immune cells that happen to be circling around to prevent local HIV infection, because if you can't go much further with the ring. So if the HIV gets beyond the vagina, the vaginal tissues, you've lost the battle. So we have chosen specifically with the peptide to target the vaginal fluids. And like I said, it's more like a shield. It prevents HIV, it coats HIV essentially and prevents it from doing anything later. So it's essentially an inactivated virus. So all the other drugs work on HIV that's replicating, so it's already you're already infected technically, but it's a local infection, whereas this will prevent any infection from happening in the first place.
Why do you have to get approval from the FDA for a product for sub-Saharan Africa?
Excellent question. You don't. You don't. But you don't. In fact, FDA in previous projects, when we came to them with the African trial that we were going to have, they said, "Why are you coming to us with this? You don't need our approval to do the trial in Africa." The problem is that there's a lot of colonialism still from a mentality perspective in a lot of these countries. So if they say, "Well, we don't want to be guinea pigs for you,"
Yeah.
and so you really have to get FDA permission, even if you're not going to do anything in the US, and get the African agencies to buy in at the same time. And then you can say, "Look, you know, we're not taking any shortcuts. This is, FDA would let us do this. You're letting us do this. We're going to do it in US women and African women at the same time, so that there's a trust issue that's not preventing you from making progress."
I have a question, but just from my experience doing clinical trials, I have found it more reasonable to deal with an African clinical trial, even as a precursor towards United States clinical trial. So that has value in that way, especially when you're talking about future commercialization.
Yes.
But my question is, one of my questions is social, and that is, why is it that women are more at risk in Africa than in the United States, where it's usually men having sex with men?
I think it starts with there's a much higher percentage of the population that has HIV in Africa. And so your risk is greater just from a probability perspective. Here in the US, the HIV epidemic is very heterogeneous. It's very focused on men who have sex with men. They're the group that really has that much higher HIV prevalence. And also receptive anal intercourse, your chances of getting HIV are much higher than vaginal. Now, in sub-Saharan Africa, it's a totally different problem. These young women are oftentimes not treated particularly gently, and so that increases their risk. They cannot protect themselves because their partner doesn't want them to, and so unless, for example, the partner won't wear a condom, and if they have an implant that you can feel, then that's already a negative. So there's a lot of stigmatization, and a vaginal ring that is not obtrusive, where the man can't feel it, and he certainly can't see it, doesn't know she's wearing it. And if he asks, you can say, well, it's a contraceptive ring, I don't want to get pregnant. And the men will know about that. Then I think it's a much lower bar in terms of getting there. I think that's probably one of the reasons why.
Okay, then I have one final technical question. How do you make the prototypes of these rings?
Well, if you take the tour, you'll see some of the machines that we have here. So we do some of the early stage prototyping by 3D printing, like most people would. 3D printing is not a viable technology for manufacturing large amounts of inexpensive products for this. We're looking at getting the price way down. So the standard injection molding techniques that exist, that have been around forever, are very good for making large quantities. And we have injection molding systems, both thermal and silicone, downstairs as well. So early prototyping we do here. John has a machine shop as well, where he makes a lot of the molds and other parts. And then we would subcontract to a manufacturing company to make us larger quantities, because we only are really set up for making small quantities here. So it's really a process.
That's why I said prototype.
Yeah.
So, there are more questions online. We can say goodbye to the online folks and maybe do a tour.
So
Okay, so
Thank you. Great talk, Mark. Great work.
thank you.
thank you. I can't see anybody online, but thank you very much for joining us. And join us next time.
If you want to do a tour, we have 1, 2,